We are lucky to have Jennifer Nemhauser from the University of Washington. Jennifer will be giving a talk entitled Plant Logic. Thank you.
Thank you. Great. So first, I'd like to add my thanks to everyone else's, to the organizers for this event, and for inviting me to get to talk a little bit about the work that we're doing in my lab. I think when I think about the question that has sort of motivated me throughout my career, it's one that maybe many of you also share, which makes it fun to be here, which is the question of how biological systems process complex information. And for me, as you might have already gleaned from the title of my talk and from this beautiful animation, the systems that I'm really interested in understanding that information processing within are plants. And there are a number of reasons why I think that plants make for a particularly exciting place to ask questions about information processing. And I'll just hit on a few of them here to get the questions that are sort of the overarching ones that we're working on in my group. So the first is really trying to understand at a molecular level how do these different components work together in a way that allows plants to process signals that have different meanings in different contexts, and that these signals are also coming from a very complex environment as well as from endogenous developmental programs that are continuous throughout the life cycle of the organism. And the second part of that, the second question, is how do we move from what's happening in individual cells to really understanding how this branches out into multicellular responses, particularly in a context where we're really talking about distributed information processing. And how does that work in an efficient and coordinated manner, especially when we might be talking about this kind of coordination that has to happen over the size of like a redwood tree, right? This is very long distance kind of coordination. And I should say, and relates to both also to metabolic state as well, which is quite complex in a photosynthetic organism. And finally, I'm really excited to try to think about how we can take what we're learning about how plants are already doing these kind of processes, how we can glean information from those studies to help us think about how we might be able to rationally engineer improved performance in these systems that, of course, all of us depend on for it to be able to prosper and do the kind of science we want to do. So one of the first really big steps in answering these questions came a little over 10 years ago. Started with a project that has initiated a collaboration that continues to today with a great colleague of mine in the Department of Electrical Engineering, Eric Clavens. And we had this idea that perhaps what we really needed to make advances in understanding the mechanisms of molecular logic was to be able to take parts out of plants, take these signaling circuits out of plants and move them into a new context. The one we started with was Saccharomyces cerevisiae yeast, to be able to really ask much more to get much more detailed information about how the dynamics and structure-function relationships of these molecular components sort of fit together. And so I would say that also we were really lucky as two assistant professors to also be able to convince the Allen Foundation to invest in us in what was at that time seen by some as a kind of crazy idea to do this kind of transfer. And so that original awarding of an Allen Distinguished Investigator Award to both of us sent us on a journey in the last 10 years that has had reaped really tremendous results that I think are continuing to pay off in interesting ways. So this is really I'm going to tell you about just one pathway in particular that we have gotten the farthest with in this process, and that is the signaling response to a hormone called Auxin, which is one of the fundamental chemical signals that plants use over and over again in all of these different contexts to do really diverse and interesting things. And one of the reasons why the Auxin signaling pathway was a great place for us to start with these studies is that it's arguably the best understood signaling pathway in plants, and it's also quite short. It has not too many components. And so Another one of the things that makes it really useful for thinking about it in a broader context in terms of all eukaryotic signaling is that the auxin response pathway, each of the components that I'll tell you about in a minute, it's composed essentially of these three modules, each of which rely on interfacing with basic cell biology that is conserved across eukaryotes. And in fact, this is why we can do this synthetic biology approach of moving things from plants into yeast and having things mostly work the same way. So this is the basic outline. It should look familiar to you, to have many things in common with many of the signaling pathways you may be interested in. And it's starting on this lower bottom right of the slide with thinking about transcriptional activation, where we have this transcription factor family, auxin response transcription factors, shown in green, that bind to the promoters of auxin responsive genes. When auxin levels are low in the cell, these ARF transcription factors are kept in a repressed state through interaction with these gold adapter proteins called OXIAs, which connect the transcription factors to members of the TEP1 Groucho co-repressor complex. The plant name for this particular family member is TPL, and it's shown in purple. So that's how the transcription is kept repressed when auxin levels are low. As auxin levels rise, the adapter proteins actually act as co-receptors with an F-box protein shown in the brightest blue at the tip of that SCF E3 type ubiquitin ligase that is able to essentially, so there's a very strong affinity of the F-box adapter, the F-box and the adapter complex for auxin, and this results in essentially gluing the adapter to the E3 in a way that you get polyubiquitination and subsequent degradation of the adapter, and that releases the repression on the transcription factors. So what we found, quite actually to our surprise, is that it only took 5 plant components that we had to move from plants into yeast in order to recapitulate that entire response pathway, so we can give now auxin to yeast and get any gene of interest to be turned on. And I've outlined just in these boxes what those different components are and also how they relate to those different modules. And being able to do this enabled us, of course, to also study each of those modules independently, and also to be able to get at the variation that is represented by the different family members that, for example, are represented by each of those components. And the kind of ways that we really got at dynamics as opposed to just this static state is by using flow cytometry and experiments kind of like the one that are shown here on the right, where we have time going along the x-axis and fluorescence on the y-axis, and by putting fusions to fluorescent proteins or in specific places in the pathway, we're able to get things like what we see here, where in an individual cell we can actually add auxin at time 0 and see the repression complex degrade and disappear, while we see a reporter that's downstream of one of these auxin-responsive promoters increase over time. And so we can use this user-defined circuits to basically go in and substitute whatever family members we want and whatever combinations and ask questions about differences in dynamics. This has been extremely useful for being able to get at some fundamental questions about things like how does degradation rate of substrates, how is it controlled, how is it encoded in both the receptor and also in the substrate. And those are things that are actually, I think, quite useful outside of even this specific pathway. We've also been able to start doing some really nice evolution studies with looking at either natural variation within these families or also across different plant species. And so all of that, I think, it really shows how having the right technology at the right moment opens up new questions and new avenues of response. And a lot of what we're doing now, we've worked a lot on the degradation side, but now we are also finding that things like what are TEP1 Groucho TLE type co-repressors doing is actually turns out to be a kind of controversial question. And this kind of system is allowing us to ask as a single locus in a heterologous system some really interesting questions about what repression really means and how it relates actually to the dynamics of activation. So I want to get back to that last question to say that one of the other that I posed on the first slide, so one of the other things that we can do by sort of studying these pieces in real detail is also ask questions about, can we use them now to essentially go in and rationally or semi-rationally engineer interesting traits. And so one of the ones that we started out with in asking this question is illustrated by this really nice drawing here that shows modern corn, a modern corn plant on the right, and to the left of it is its wild ancestor, teosinte. And you can see there's a lot of different things that have happened over the course of domestication that to change the form of teosinte into corn. But I think that one of the most obvious things about it is a decrease in branching. And this is a really interesting phenotype because we know there's actually quite a lot of good evidence here to say that being highly branched, like the wild teosinte, has a lot of fitness advantages for a wild plant. However, in any kind of crop, the degree of branching is actually should be as low, as little as possible, and that's because actually one of the ways in which farmers most efficiently increase yield on of their crops is by densely planting their fields. And so the more tightly you can pack your plants, the more yield you are going to get. And so being able to, for example, go in and rapidly change the branching pattern of a heritage crop for a subsistence farmer in a fairly easy and directed way, rather than the 10,000 years that it took to go from teosinte to corn, has real consequences for our ability to potentially help increase food security globally. So this is also, as I said, a process that branching itself is a process that's been studied for a long time, and we have a lot of information about the molecular mechanisms by which it works. And this is actually the cartoon shown here, taken from a really nice modeling paper that kind of illustrates the very core feature of how it is determined which of the branches actually grow out in a plant. And what you can see here, the core of it is essentially how effectively an individual cell, represented by this box here, is able to get a polar localization and accumulation of an Auxin efflux carrier. And so these cells that are able to essentially canalize the flow of auxin out of the cell are the ones that sustain growth and grow out. And so you can see in this situation where you see a competition between an apical and lateral bud, the bud that is actually able to efficiently cause this polar localization of auxin, of the auxin efflux carrier, and in that way become an efficient exporter of auxin, that bud is able to grow, whereas the lateral bud remains dormant because it cannot. And so this positive feedback loop between the level of auxin and its the expression and activity of its efflux carrier is the sort of critical point for determining flipping between these two states. So our question was, can we go in and use this information to think about a way to engineer it? And so in order to do that, we came up with a new technology that is illustrated here. And we relied on, it was actually the original instantiation of this was something that a really talented graduate student, Arjun Kakar, first thought about doing in terms for engineering cell-cell communication in yeast. So this was originally done in yeast, and then he took it into plants to get at the questions that I'm talking about. And the core of this technology is shown in the middle of the slide. It's a modular programmable transcription factor that's controlled by Auxin, and it's comprised of three subunits. One is a Cas9, so a catalytically inactive Cas9 that can target this protein to DNA in a programmable way, depending on the guide RNA provided. That's fused to a sequence that's necessary and sufficient for auxin-induced degradation. And finally, there is a repression or activator domain that is further fused to these proteins. And the ones I'm going to be talking to you about, we used that TPL, that purple protein I showed you on the original model, middle, the plant repressor domain is fused to the end. And so Arjun actually termed these hormone-activated Cas9-based repressors, or HACRs. That is one of the benefits of bringing engineers and biologists together. Is, I wanted to name them spiders for reforming the web, and he wanted to name them HACRs because of thinking about reprogramming the developmental programs. And so, of course, Arjun won out. So because we are entering the age of engineering, in case anyone is wondering, I think that is absolutely the case. So the way that this technology works is we basically, as I said, we have this transcription factor, this HACR that is targeted to a promoter. It's itself constitutively expressed. The guide RNA is also constitutively expressed. And the third constitutive promoter is driving expression of, in this case, any gene of interest, and in this case, a fusion of the venous luciferase reporters. And so in the way this logic works is when auxin levels are low, this transcription factor is keeping the level of this level of expression of this reporter low. And when auxin arrives in a parallel to the native auxin response signaling system, this whole protein gets turned over, targeted for degradation and turned over, releasing repression and you have activation and expression of the reporters. So this, if we put all of those components into plants and apply auxin exogenously, we can see that indeed we can get nice induction of expression of venous, or in this case, looking at a dose response, or a time course rather, of luciferase activity, we can see that after following addition of auxin, we get a nice induction of luciferase activity that's dependent both on adding auxin and also depends on having a functional degron. So if we stabilize the degron so it's no longer auxin sensitive, you can see that you don't get a response. So this technology itself is only useful if it's actually also able to be responsive to the dynamic range that is present in planta. So the differences in auxin levels that you would actually see during a developmental program being initiated. And so we added an additional feature to this of make it a ratiometric line, a ratiometric sensor, where we have the same promoter that's driving the HACR targeted, that is normally HACR targeted, but we've made a mutation in the guide RNA target site and have that driving a second fluorescent reporter. And so in this way, we can account for any differences, small differences in expression pattern of the promoter. By using a ratiometric output. And we can look for things where we know that auxin accumulation occurs, and we've looked at this in many different tissues, and I'm just showing you one example here, where we have a mature root that has relatively low auxin levels, but we know one of the very first things that happens upon production of, or initiation of a new lateral root is a auxin maxima. And we see very nicely that we can see at very early stages that auxin accumulation, and as the root starts to grow out, we continue to see a persistence of that auxin maxima, as has been visualized with other reporters. So we feel really confident that we've seen this in lots of places, that where auxin is normally, the different levels where we see low auxin minima and maxima in native contexts, our HACR construct is able to respond. So the next question is really then getting back to this, can we then go ahead and attach this HACR now as a mechanism to essentially alter the gain in the system in that feedback regulation that I told you about? So the more auxin you have, the more efficient you are at producing auxin efflux carrier. Can you essentially switch plants from having a strong canalization feature, which is more like the wild type plants, where it is fairly easy to get canalization going and where you get buds to grow out? Can you switch that by adding this drag on the system with the HACR to have a more crop-like weak canalization where those lateral buds get repressed? And the answer to that question is yes. So if we target our HACR construct, if we add a guide RNA that targets the PIN1 promoter, what you would predict to happen is that now there would be this drag. You would need much more auxin around in order to get thank you, in order to get canalization to be kicked off. And so you should see less branching, and indeed that's exactly what you see as seen in this picture and also seen in the quantitation on the right. So that's very exciting. And what is perhaps even more exciting to us going forward is that this is, as I mentioned, this is both a programmable and a modular system. And one of the nice things is we can swap out that degron domain for anything that is turned over in response to a signal in plants or likely in any eukaryotic system, I would say. And so we've done some extension of this with looking at things like substituting a sequence that is responsive to a different plant hormone called jasmonate that is turned on in response to damage caused by chewing insects. And so what you can see here is that essentially looking at one of these luciferase assays, after leaves are damaged, you see that in the leaf that is damaged, you get nice induction of the reporter. You don't see that in the control plants. We've now hooked this up to expression of the Bt toxin and are in tests now to see if we can actually get nice resistance to chewing pests through this inducible response, which it has the added benefit of only producing the Bt where it is needed, which decreases metabolic load and likely also keeps it from being expressed throughout the plant in things that we may not, in parts of the plant that we may not want, where we may not want it to be expressed. So, in summary, I just want to quickly say that this technology is allowing us both to really get access to parameters in the models that many of us are interested in. So being able to actually modulate feedback is a really important factor in being able to understand how biological input-output systems really work. And it is also has this engineering potential for being able to access new parts of design space. And I'm really excited about that, especially thinking about what kinds of plants we may need in very rapidly changing climactic environments. So with that, I just want to briefly say thanks to Arjun Kakar in particular, who's the graduate student who really spearheaded most of this work. Andrew Lemix is an outstanding undergraduate who's taken on most of the work with the jasmonate HACR. Alex Layden is a postdoc who is also helping out with this project and taking it into some new areas with different degrons. Eric has been my longtime collaborator, and I'm very grateful to him, also to our funding, and to all of you, and I'm happy to take any questions.
Thank you. Let's have some questions.
That was really fun. I'm curious why you had to export the system to yeast and why you couldn't sort this in plant cells.
Yeah, great. So, right. So the problem, so there's a number of answers to that question. One of them is that auxin, for example, is present at some level in many, many, many cells, basically. So it's very hard to have a base state. So unlike a lot of situations in animal systems, which I really appreciate and envy, where you can actually start with a naive cell and then apply a signal of some kind and then watch what happens, the cells that we're looking at are sort of always in constant, there's constant auxin flux around them, and there isn't really much of a naive state. So it's hard to ask, going from base to induced, what you're really, it's hard to find a place in the plant to look at that. The other issue for us that's probably even a bigger issue is that these are all part of very large gene families, all of these components, and they're all co-expressed in the same cells. And so it's very tricky to ask questions about what is the role of an individual part of the pathway in an intact system. And so by moving it into a system where we actually control all of the parts, that has been really important. It is also the case that the auxin system, I mean, one of the things that is really a feature of having a very few number of chemical signals that get used over and over again is there's an incredible level of co-regulation and cross-regulation across these pathways. And so it is also it is one of those situations where every time you are perturbing something, you don't know exactly if what you're looking at is direct responses in that system or actually a huge amount of compensatory responses coming through other systems. So that's some of the reasons why it's been really important to have a heterologous system to look at.
Thank you. We have time for one more question.
Conversely, can you take advantage of the yeast system to identify temperature-sensitive mutations that you could put back in your plants? Because then you could control the cannulation at different temperatures, which could actually be beneficial for growing in adverse climatic environments.
Yeah, absolutely. We've been exploring all sorts of ways in which it has been really fun to see how well actually the behavior of things in either engineered natural variants that we have in the yeast system, when we put them back into plants, almost always work as predicted. And so when they don't, that's interesting also. But yes, it is absolutely given this platform for doing really rapid evaluation. And in the plants that we, the model plants we work in the lab, it's not such a time delay issue. It's not such a hard thing. But when you're talking about trying to engineer cassava or something that's slow growing, difficult to do, this kind of being able to have a system to do rapid prototyping is really important for before you go back into those plant systems. Yes. Absolutely.
Thanks very much.